Hello, my name is Dr. Eric Landrum from the Department of Psychology at Boise State University, and this is my research method screencast on types of validity. Now, there are different systems and ways to think about validity. I'm going to present just one of these during this screencast, but I do want you to know that there are other ways to think about validity. For example, if you were taking a psychological measurements course, you might think about construct validity, content related, criterion, face validity, but actually I'm going to take a little bit of a different approach in today's screencast. So here we go. So the approach I'm going to take is really going to present four different types. I'll give you an overview of each of the four, and then we'll dig a little bit deeper in some of them, especially digging deep in the internal validity section. But to get us started, statistical conclusion validity is the validity of inferences about a correlation or covariation between treatment and outcome. So if we're expecting a cause and effect relationship, first off, do the variables seem statistically related? That is, if we were to expect that height was a causal factor of weight, we would expect them to be significantly positively correlated. They are, but that's not in and of itself enough to demonstrate a cause and effect relationship. And so when we expect one event to cause the onset of another event, we expect to find some statistical relationships when we look at those two events together. And so that's one of the types of validity that we want to be looking for. Another one, and this is probably the classic of the four that I'm going to talk about today, is internal validity. So we're addressing the validity of the inferences about whether the observed covariation between some event A and some outcome B, is it causal from A to B? So is the relationship causal? So in the example I just shared with you, there could be a significant positive correlation between height and weight, but it doesn't make sense that height causes weight or weight causes height. And that's one of those things we talked about way back in the critical thinking screencast about third factors and plausible alternative explanations and whether something is descriptive or quasi-experimental or relational or that top rung of the ladder causal. Here's a Mark Wahlberg theme, and I like to embed these whenever I can find them, and they're related to the content, of course. And so internal validity talks about we think that one of these is causing the other one of these. Is that really the case? Or could there be a host of other third factors that are masking what's really going on? And if we dig a little bit deeper, we'll find the true relationship between A and B, or the intervention and the outcome, or the independent variable or the dependent variable. And again, this is just a little bit of an overview, and we'll dig deep into this area of internal validity. Another type, and you might hear this again if you were talking more about psychological measurements, a questionnaire construction viewpoint, is construct validity. This is kind of the 30,000 foot level of validity. Just because we say we're measuring something, are we really measuring the essence of that construct? And so, for instance, way back to the critical thinking screencast, we talked about an operational definition of hunger. And so I could ask someone about their hunger level on a scale of 1 to 10. I suppose I could weigh them. I could take a blood sample and look at glucose levels and make an inference about hunger or not. But construct validity tackles this by looking at, did we measure what we really thought we measured? And so did we measure hunger, or did we measure some proxy for hunger that may or may not be that directly related? One of the ways that we determine this is by conferring with content experts or looking at other studies and comparing our results to previously established metrics to see if we can have some concurrence in the outcomes and see if we can build that case for construct validity. And finally, in our quartet of types of validity that we'll talk about in this screencast is external validity. And so this is about the inferences from the observed cause and effect relationship. Can they be generalized to other people, other settings, other treatment modalities, and even other variables? So in other words, can we generalize the results of the cause and effect we found in our study to other people, places, and times? Is it generalizable, or is it so specific to the participants in the study that there is not much nomothetic value? That is, we're not going to be able to generalize from the smaller group to the larger population. We're not going to be able to draw an inference from the sample back to the population. All right, so that's our basic overview of the quartet. Now I want to kind of circle back and talk a little bit about each one in turn. But again, we are really going to focus eventually, or most of our effort, on internal validity. Now the consideration of this topic is important because we want to avoid these confounds. When we conduct our experiment, we want to test the effects of the independent variable on the dependent variable, and we'd like to avoid this notion called a confound. A confound is a result that has been an influencer or influenced by some systematic but unspecified factor. So a confound is going to be something that happens maybe during the study, unknown or known to us at the time, that might be influencing both the effectiveness of the independent variable as well as the accurate measurement of the dependent variable. So to the extent we can, we want to use our research methods to avoid or at least minimize the impact of confounds. Another way to say this is that a confound is part of the results of an experiment are not due to the manipulation of the independent variable, but maybe some third or fourth or fifth factor, some hidden or latent aspect of the study that we didn't realize. And this may sound vague right now, but give me a couple minutes and I'll try to give you a bunch of really concrete examples, especially when we're talking about internal validity. So a confound essentially is anything that systematically biases the results of the research. We understand that randomness is going to occur and there may be random effects. We understand we're going to make errors when we draw conclusions, but a confound is this systematic bias. For example, it might affect one group more than another group, that would be bad, or one variable more than another variable, that would be bad. We understand there'll be randomness across the study, but we don't want any systematic bias creeping in. Otherwise, we're going to draw a conclusion that's going to be inaccurate, and that could result in either a type 1 or a type 2 error. So as I kind of circle back to those four, and we'll do that in order, a threat to statistical conclusion validity, we talk about where we might look at a correlational relationship and the conclusions that we draw may be incorrect. And so these are threats to that. So for example, if you have low power, sometimes you can capitalize on chance a little bit more than we'd like to, and that's going to lead to conclusions that may be erroneous in nature. If we use statistical tests in a way that they weren't designed, and so every statistical test has a set of assumptions about the normality, the distribution of participants, the scale of measurement it's accurate and appropriate for, and the more of those assumptions that we break or violate, the more of a threat we're going to have to the statistical conclusion validity of our study. Phishing and error rate can be a problem too, and what this really means is that dipping back into the same data set over and over and over and reusing things. Every time you conduct a statistical test, there is a chance of error. That is to say, when you're going to draw an inference from a sample back to a population and use an inferential statistic, there's a chance of making a type 1 error. And so we want to minimize the chance of making errors by minimizing the number of potential conclusions that we draw. So phishing and error rate can be a problem. So this is why it's really important to have a detailed, specific hypothesis that closes out your introduction section so that you know what you're going to be testing for once you have your data in hand. We don't want to just keep doing statistical tests until we find something. That would be an inappropriate approach to take. And also unreliability of measures. And so if we're unclear that our depression test is really a good test of depression, or our survey about self-efficacy or our definition of college student success isn't well defined and measured, then we're going to have problems in drawing conclusions from the data analyses based on those perhaps faulty measures. And so we're only as good as the measurement devices. And so the more accurate we can be there, the more representative they are of the true constructs, the better we're going to have a chance to identify meaningful relationships or differences between variables or conditions. Internal validity, I suppose, is the granddaddy of this presentation of the quartet of different types of validity. And so I want to kind of get into this classic list. When internal validity is present, when things are working the way they should, the independent variable is affecting the responses to the dependent variable while all else is held constant. And so in the ideal situation, we design a weight loss study and the intervention's working, the control group is not changing, and we can show the beneficial effect of the intervention, the treatment modality, whatever you want to call it. So when it's internally valid, A is a clear cause of B with no other competing explanations as to why B happened the way it did. That would be a rare and unusual event to never have a threat to internal validity, because people are human and experimenters are human, and humans are random at times and not perfectly controllable even in the best laboratory situation. Really, in essence, what the question about internal validity is asking is there on the screen. Is what you found really what you think you found? Is what you found really what you think you found? And so it's important to be confident that the relationships that we identified not only are statistically appropriate, but also have this internal validity. They make sense. There's a clean causal chain between the event and its outcome. So I want to present to you briefly and somewhat quickly, however, this classic listing of threats to internal validity. So what I'm about to present here in this threats, these are really classic confounds that might interfere with the strength of our conclusion between the independent variable and the dependent variable. And by the way, we don't want that. We want to avoid confounds. We want a clean, clear test of the impact of the independent variable on the measurement of that dependent variable. So again, these are confounds. These are threats to internal validity. So history is a classic one. So any event that occurs during the course of the experiment other than the independent variable that might have an impact on the participant's behavior. So a classic one may be that you're in a classroom taking a long survey and you're filling this out, and all of a sudden the alarm goes off in the building, the fire alarm, or the power goes out. It's an event that might happen that impacts how you're reacting to the dependent variable measure, but really isn't part of the study. So this event over time, this historical event, may be a threat to the conclusion that we would draw from the independent variable influencing the dependent variable. Another threat is maturation. So any systematic changes that occur within the person, the participant, during the course of the experiment or as a function of the passage of time. So in longitudinal studies and studies that take a long time to complete, this could be a problem of interpretation because, and as you can see a nice list here, the participants become older, wiser, stronger, healthier, tired, bored, fatigued, invigorated. And so this might be over a 10-year longitudinal study. People change, and you're looking at their intelligence levels in a career, but they decide during year 5 that they're going to go back to college. They learn more, they gain skills. By year 10, they're back in their job. It's going to look like a change in the job, but there was really something else that happened along the way. There was some intellectual maturation, if you will. Something happened during the study that changed the pattern of responding that really wasn't designed to be part of the study. And so this is just a threat to longitudinal studies that over time things may change that are not part of the experimental design. And so that's going to be something that you take into account. And usually in longitudinal studies there's a large battery of items that are asked at the very beginning of the study and at different checkpoints along the way, so that you can hopefully rule out those alternative explanations for changes over time and hopefully demonstrate the relationship or the variables or the differences that you're looking for in your longitudinal study. Another threat to internal validity is called test sensitization. And actually, way back in an earlier screencast, we talked about this a little bit. This is the sensitizing of participants to relevant variables or the opportunity to practice skills via a pretest. So This is a problem with repeated measurements of the same individual because folks may become sensitive to their own behaviors and learn from their own prior experiences. In fact, we call this a threat to internal validity, but actually, outside of a laboratory setting, this is exactly what we want people to have take place, right? If you attempt something and you fail at it and you learn from that and you change your behavior the next time, we actually call that learning, and that's a good thing. But in the course of an experiment where I'm trying to have this controlled situation where the independent variable is affecting the dependent variable, test sensitization actually turns out to be a confound. So in other words, if I was doing that GRE, that graduate record examination, and I had you take a pre-test, and then I have you take my test preparation course, my prep course, and then a post-test, and I had a control group that took the pre-test and the post-test, you might actually learn something from the pre-test that sensitizes you and changes your behavior when you take the post-test. In fact, we'll be able to detect that in a pre-test, post-test control group design because we've got that pre and post without the test course. So now we look for, does the test course lead to differential gains above and beyond what we see in the control group? So I just want to reiterate, in the normal setting, this is exactly what we want to happen to human beings. We want you to be sensitized to past experiences, learn from them, adapt and adjust your behavior appropriately. But in an experimental design, in a research study, this actually might muddy the waters in figuring out, is it just the repeated measure effect, or is it the repeated measure effect plus whatever impact my independent variable is having? So it does it's a threat that kind of muddies the water in figuring out that causal relationship between A and B. Another one that's one of the classics in this list is called statistical regression threat to internal validity. And this talks about the movement of extreme scores towards the mean. If you start at the extremities, you will move towards the mean over time without any intervention at all. It's almost the reverse of a ceiling and floor effect. If you start at the very top, there's nowhere to go but the toppy top. And so if you start at 100% on a scale of 0 to 100, the only direction your grade can change is down. By the same token, if you started at 0% on the first test, the only direction you can go is, I suppose, stay at 0 or go up. And that notion of when scores start at the extremities and they move toward the mean is called statistical regression, or sometimes you'll hear it called regression towards the mean. Sir Francis Galton, a relative of Charles Darwin, actually studied this phenomenon when he looked at intelligence levels, even height of fathers and sons over generations. And what he found is that if you start at the extremes, you tend to move toward the middle with regard to intelligence, which he labeled there in quotes the principle of mediocrity. So sometimes the scores might be changing, but it's not because of the intervention or the independent variable. It's because where the scores started on a scale. And so you can see it kind of graphically here. The top curve, if you have a normal distribution, and you start with the circles, if you start with the lowest of the low scores, and over time with a generation, or you take another test, you start with a really low score, odds are the next test score will move towards the middle. It could only stay at the very bottom or move towards the middle. And the same is true with what's on the right there. If you start with the very highest level, you take a test again, you could either stay high or move toward the middle. When Galton did this, he did it with intelligence of fathers and sons. And so extremely intelligent fathers tended to have sons that weren't as intelligent, not really so much genetically, although he would have made the case genetically, but there was nowhere else to go. If you start at the top of the scale, you can either stay at the top of the scale or move inward, move towards the mean. That's called regression towards the mean, or a threat called statistical regression. All right, we're moving down the internal validity threat hit parade. Selection is another threat to internal validity. This is the possibility that the experimental and control groups are not equivalent prior to the experimental manipulation. This is bad. We don't want this to happen. We don't want the groups to differ initially from one another except for random chance. And so think back to that graphic where you saw the circles in the bubble, and we extract we extract the sample out of the population, and then we randomly assign the sample into two groups, experimental group and control group. But this is the problem of those two groups, the experimental control, are not different from the beginning. So we used our random assignment, but it wasn't effective. It didn't work. In fact, there's a difference that exists before the study even starts, before you start the independent variable manipulation. And so this is a problem because I might find a difference at the end of the study, but it was really there all along. My independent variable didn't have any effect, but it was because of this threat, this selection artifact, that I'm not going to be able to draw the conclusion that I want. Or I'm going to draw a conclusion, but it's going to be a type 1 error. I'm going to say that the groups are different, but they really weren't different. It was because of this type of error. So self-selection or volunteerism is a real problem here. And so sometimes in some research scenarios, you have to rely on volunteers, but if at all possible, you want to randomly assign volunteers to either the treatment group or the control group. That way you can minimize this self-selection bias that might creep into the data and then complicate the interpretation of the results of your study. Another threat to internal validity is called instrument decay. So this is about the recording devices. Any decrease in the accuracy or sensitivity of the recording techniques during the course of the experiment. That is to say, changes that occur in the measuring device. So this might be a glitch in the computer program. This might be you're doing an online survey and there's a glitch in the logic and you can't finish correctly. In the olden days, this might have been bubbling in a bubble sheet with a number 2 pencil and poking a hole in the bubble sheet, and the scanner had a hard time figuring out what the responses were. So this is about changing the responses of the participant, but not in a way related to the goals of the study. This is because of some instrument failure, of some instrument, some methodological flaw that happened that was unknown and unplanned. That is to say, it was a confound by the time the study was concluded. Another, and this one sounds way worse than it actually is experimental mortality or attrition. This is the loss of participants during the course of a study or an experiment for any reasons, but it's particularly troubling when participants drop out of one group compared to the other. So let's say in our classic 2-group design, we've got our experimental group and our control group. And 75% of participants drop out of the experimental group, but only 4% drop out of the control group. That is a threat of experimental mortality or attrition. Over the course of time in a study, we expect some loss. That is, people will sign up for something but not complete it. We don't expect a 100% response rate. We don't expect that everyone that starts a survey that takes an hour is going to finish a survey an hour later. There's going to be some dropout, but we want the dropout rate to be relatively the same for whatever conditions of the study exist. And so and a lot of times you'll see this in a weight loss study if it's not taken care of carefully. So for example, if you're in the experimental group and you're losing weight, it's working, people are motivated to stay in that study. But if you're in the control group in that weight loss study and you're getting some sort of placebo and over time you're not losing weight, you might go, oh, I'm going to forget it. I'm going to drop out of the study, or I'm going to stop recording my weight once a week like they asked me to, because they get depressed about the study isn't working. And they might even actually figure out that they're in the control group. And so if one group drops out at a different rate, at a substantially different rate than the other group or groups in the study, that's when we have an attrition problem, and we need to address that and see what happens. Sometimes you see this in survey research with non-respondents. So if I do a survey of 1,000 people and I get a response rate of 4%, I've got to worry about what the 96% of my non-respondents are feeling about the topic compared to the 4% who did. Did the 4% who stayed in the study have an axe to grind? Are they overly positive, overly negative? All of a sudden now I've got competing alternative explanations they have to worry about when I'm trying to draw my cause and effect conclusion, or even my correlational or relational conclusion for that matter. And finally, and I think this is the last one in this list of internal validity, ambiguous temporal precedence, a very fancy word or series of words that talks about if I'm trying to establish cause and effect, I need a time sequence. The cause has to occur before the effect to have a cause and effect relationship. And so sometimes there may be a lack of clarity about what event happened first. Did the change happen, then the independent variable manipulation, or the independent variable manipulation was clearly before the dependent variable observations took place. So, for example, if you're looking at weight loss and self-esteem in teenagers, is it that the weight loss happened and that helped us feel better about our bodies, or that our self-esteem went up in some way which helped us lose weight and feel better about our bodies? And so sometimes it's really clear in a true experiment, but if you're doing a field study, if you're doing if you're doing a quasi-experimental study where you're just looking at variables as they exist in nature, it may be actually very difficult to disentangle the order of what caused what. So it's good to know the relationship, don't get me wrong, but if we have ambiguous temporal precedents in our midst, that means that we're going to have a hard time establishing that internal validity that we want for that cause and effect relationship. All right, let's take a quick peek here at the last of the four versions, external validity. And external validity, again, as a reminder, is that ability to generalize the results from the sample to greater populations. And so maybe it's a sample of 200 students that are going to be generalized to 20,000 undergraduates at a college or university. Or maybe it's a college or university sample that's generalized to the broader population in the state or the country, the region, the world. And so external validity talks about what we're going to do with the results and what interpretations we're going to make beyond the confines of the study. That is, to the extent to which they can be generalized. And so to be careful with our external validity, we have to examine the subject variables of our participants. And remember, subject variables are those traits, qualities, and characteristics that we each carry around with us. And so age, gender, occupation, education, socioeconomic status, personality traits, marital status, so on and so forth. So the better the match the population to the targeted let me take that back. The better the match from the sample to the population on these traits and qualities, the stronger the generalization could be. So if I'm really interested in generalizing from my sample of 200 to my college student population of 20,000, and if I know age is a critical variable, I'm going to want to look at the age of the sample and the age of the population. And if those are close, then that's a stronger case for generalizability. The further apart those two ages are, the weaker the case it's going to be that I can generalize from my smaller sample to my larger population. Also, when looking at external validity, we want to examine the generality of the research, the nature of the researchers, how it was conducted. Was it a lab study? Was it in the field? Is it out in nature? Observational study? Incidental events like the time of day. Sometimes if you do a study with college students, the time of day can make a huge difference, whether they have to show up for an 8 a.m. session in a classroom or 4 p.m. session could make a world of difference in generalizing the outcomes of that study. Also, levels of manipulation in terms of the independent variables, even measurement operations, the modality. Is it on a computer screen? Is it going to be paper and pencil? Is it a web-based? Is it app-based? Is it smartphone? The way that we collect the data and all the details of that methodology can have an impact on the level of generalizability we can have, again, from that sample to that population to be able to draw that cause and effect inference. All right, that's it for me, and a very brief overview about validity and its threats for research methods.